Hello, welcome to the second video of biochemistry, chemistry 3111. So in this video, we are going to answer questions for the past paper for 2024, which was test 1 for biochemistry. So the first question says, identify which level of protein structure is represented by each of the following diagrams below. So this is question A. So as we can see here for this structure here, we have two things here. We have the beta pleated sheet and we have the alpha. So this basically means that this is the secondary structure, because secondary structure is made up of alpha and beta sheets. So this is, for one, it's secondary structure. So for the next one, we have this one here, which looks more like a rope. It's long, it's elongated. So this one, as you can see, we have a lot of ball-like structures, which are just the amino acids that are attached together in a long sequence. So this is just the sequence of amino acids. So this structure here, it's a primary structure, which is just the sequence and the number of amino acids in a polypeptide. So this is a primary structure. Primary structure. So let's go now to the third one. We have it here. This one here. So this one, as we can see here, it's more like a polypeptide that is being folded to make a 3D structure. So the structure or the level of a protein that is represented by this structure is what we call the tertiary structure. So the third structure is the tertiary structure that has to do with the 3D structure of a protein. So we have the fourth one. So for the fourth one, as you can see here, we had here the third one, which was the tertiary structure. But here we can see we have something like the tertiary structure being like there are two together here. It's more like they are polypeptides that are organized together to form a functional protein. So this is basically the quaternary structure. Quaternary structure. So this is the quaternary structure. So let's go to the next question, which will be B. So what does question B say? So question B says, what type of bond is responsible for a protein's secondary structure? So as we said earlier in the first question, when you look at the secondary structure, you only find the alpha and beta pleated sheets. So in these structures, you only find hydrogen bonds. So like in the alpha, you have the intra-chain hydrogen bonds, but in the beta pleated sheets, you have inter-hydrogen bonds. So the answer here is hydrogen bonds. So let's go to the other question, which is C. So C says, mecaptoethanol breaks which bonds present in the protein. So this chemical here, the mecaptoethanol, is just the bond that breaks the disulfide bonds that are found in the tertiary or quaternary structure. So this one just breaks down the disulfide bonds, and disulfide bonds are just formed by the oxidation of two cysteine proteins. So this is disulfide bond. Disulfide bonds. So let's go now to the other question, which is D. So what does question D say? It says, explain why hemoglobin is classified as a protein having quaternary structure. This is D. So we already discussed about this one. A quaternary structure is just polypeptides that are organized together to come up with a functional protein. So as we can see, hemoglobin is quaternary structure quaternary structure because it's a tetramer. It is a tetramer. Of 4 subunits. Subunits where you have two alpha and two beta subunits. Organized together to make a functional protein. Functional protein, which is hemoglobin. Let's put it in brackets here. It is hemoglobin. So this is all for question 1. Question 1 ends here. So let's go now to the next question. So this is the next question for question 2. So let's write question 2. Question 2. A. So here it says a group of chemistry 3111 students, which is biochemistry, wish to derive the amino acid sequence of a purified peptide. Your research technician has performed multiple tests and presents you with the following data. So the first data we have limited carboxypeptidase A digestion, and this one which produces alanine, which in short means that it cuts the C terminal of alanine. Then the second one we have the Erdmann degradation, which was done with just one cycle. So this Erdmann degradation just identifies the N terminal of an amino acid. And you know that when you want to read the sequence of amino acids, you always start from the N terminal, meaning this his which is the N terminal will be the first amino acids from the N terminal. Then we have the third one, chymotrypsin digestion, which yields free tryptophan and alanine. So this basically means tryptophan and alanine, they are separate, meaning they are different fragments, as well as one peptide comprised of alanine, tyrosine, and histidine. So this basically means that this is a polypeptide. This is one fragment. So from chymotrypsin, that means that you produce 3 fragments here. Then the next one we have elastase digestion, which just yields a tripeptide and a dipeptide. And we have to know that this elastase cuts those neutral small amino acids like valine, alanine, glycine, and serine. So let's do this now here so that we start analyzing the polypeptide. Then the first question now says, which of the following listed below best represents the unknown peptide? Let's go to the answers now so that we start. So I mentioned to say this one, carboxypeptidase A digestion cuts the alanine. And alanine, you need to know that it's always found at the carboxy end of an amino acid. Carboxy terminal, C terminal. So carboxy des A produces alanine, and alanine should be found at the C terminal. So alanine should be like this. So it has to be at the end of the C terminal. C terminal. Then the next one we have Erdmann degradation. So this one here, this histidine here, it should be at the first amino acid, which is the N terminal. So Erdmann degradation. So this one we said it produces histidine, and histidine should be at the N terminal. Know that N terminal is like this. So this is histidine here. So that's how it should look, meaning N terminal. And at the N terminal end. So the other thing, we go to the next, which is chymotrypsin. So chymotrypsin, this one, it cuts aromatic amino acids. After aromatic amino acids, so aromatic amino acids, we have tryptophan, TRP, and we have tyrosine, TYR, and phenylalanine, which are abbreviated like this. So this chymotrypsin, it cuts after these amino acids, which are aromatic. In short, they have a ring. Then it says chymotrypsin yields free tryptophan and alanine. So the results here, it yields free tryptophan, and the alanine, and also a dipeptide is alanine, then tyrosine, TYR, and the histidine. This basically means this one, they are part of one fragment. These, they should be together when you start analyzing the polypeptide, and these, they have to be different. So this tells us to say, in the sequence of the polypeptide that was being analyzed here, we have a total of 5 amino acids. So a total of 5 acids. Amino acids, just write abbreviations like this. 5 amino acids in total. Then let's now start checking on the answers, so that we start disqualifying the answers that are provided here. Because we have already known to say histidine should be the first amino acid at the N terminal, and the alanine should be at the end, at the C terminal. So let's see now the first answer. So here, as you can see, at the N terminal we have tryptophan, which is wrong. This is wrong. Can't have tryptophan at the N terminal. So we disqualify this answer here. Let's go to B. N2 alanine. As you can see, we said we discovered to say the amino acid that we need to have at the N terminal is the histidine, and here we have alanine. So this means that this answer here, we disqualify it. Then let's go to C. NH2, we have histidine here, which is the amino acid that we need at the N terminal. And at the final end, we have alanine, which is what we need. So let's leave this answer here, since we have histidine and the alanine. So this answer should be one of the possible answers. So let's go to D. NH2 alanine. Alanine, we don't need it to be next to the N terminal, so we disqualify D. So let's go now to E. So we have histidine, we have alanine at the C terminal, and we have histidine at the N terminal. So these C and E are the possible answers for this question here. These are the possible answers that we expect for this question. Now let's go to check on chymotrypsin, because we discovered to say alanine, tyrosine, and histidine, they have to be together when you cut using chymotrypsin. Now let's see E if we cut here. So chymotrypsin will come let's use red here so that we see where chymotrypsin can cut. So chymotrypsin can cut after the tryptophan. So let's say here it will cut here. Let's use red. Can cut here, then it can also cut here, so that you have this fragment here, tyrosine and also alanine. So as you can see here, the question says here, chymotrypsin yielded TRP alone, which is not what question E suggests. I mean answer E suggests. And the alanine should be alone. Yeah, E suggests it should be alone when we cut. But what about alanine, tyrosine, and histidine? So here, as you can see here, we have histidine, alanine, and the tryptophan, which is not what is on the amino acids that were yielded here. So we disqualify answer E. So which means we are just left with the option C as the correct answer. So as you can see, when you cut using chymotrypsin, you cut this one here. You are going to cut after tyrosine. When you cut here, you have histidine, alanine, and tyrosine, which is what was suggested here as the yield. Then you cut also after tryptophan, since you are cutting after every aromatic amino acids. When you cut after tryptophan here, you have tryptophan alone. I mean here, you cut after tyrosine. You also cut after tryptophan. So you remain with the alanine alone, tryptophan alone, and this one, this long fragment, which was a tripeptide that was mentioned, which is histidine, alanine, and tyrosine. So which means C was the correct answer. So C was the answer, which is NH2. We have histidine, then we have alanine, then we have tyrosine, TYR, then we have TRP, then we have alanine, then at the end we have the C terminal. So that's all for the first question, which was C. So this question had 2 marks. So let's go to question B. Question B. Provide a logical explanation for your answer in question 2. So here, this is the logical explanation that we have provided here. Let me even circle it. Or let's just write another to follow through. So this B. So the reasoning should be like this. So we are going to disqualify answers A, B, and C. A, B, and C disqualified. Since they don't have histidine and alanine at the N terminal and C terminal respectively. So the reasoning for E, the reason why it was disqualified. So for E was disqualified as it doesn't yield alanine, tyrosine, and histidine. Also, tryptophan and alanine separately. When you cut with it, chymotrypsin. Peptide was used the right fragments. When cut with chymotrypsin. When cut with the chymotrypsin. So this is all for this part here. So let's go now to question C, which says, what is the biochemical basis for prion disease? You also have to indicate the effect on normal function and list one major symptom. So let's just rub here. So what's the biochemical prion disease? Is folding of prion proteins. T prion proteins into S, which form aggregates. The biochemical basis for this, the normal prion proteins needed to form this one, which causes disease. One is made, it converts proteins to be the disease prion protein. So that's, this one is just a this one. So let's go to the question. What does the next question say? The normal function, the normal cellular function. Cellular function and causes neurodegeneration, which is just the damaging of brain cells, leading to neurodegeneration. So let's go to the symptom now for this disease, which is the next question. What's